So having come here, I don't need to convince you why we should do protein measurements in single cells. But nonetheless, I think it's useful to begin with the questions that motivate us, and in particular, trying to understand biology. Here is a piece of tissue, in this case, tissue coming from kidneys, glomeruli. And you can see that there is some structure to that tissue. So if you were to grind it up and do a conventional measurement, you're going to measure a population average level for all of the molecules present in these intricate structures, and therefore we cannot discern them. We cannot see the differences between the cells and the different extracellular matrix proteins that make up this tissue. So we want to see it in colors, and we want to be able to analyze not only the different cells as being distinct, but also want to analyze the protein environment around them, the extracellular matrix that is helping shape their phenotypes and physiology. And we don't want to see it just in 3 colors. We want to see it in full depth. We want to see it in thousands of colors. We would like to be able to measure all of the proteins that make up these distinct cells, and not just measure the proteins, but we want to understand what are they doing, what is the biology of this tissue. Measurement is just the beginning. Understanding is what's really driving us. And this meeting serves a very important role, multiple roles, but one of the most important ones that I want to emphasize here is being a Rosetta Stone for people coming from different communities, from mass spectrometry, biology, computational biology, medicine, so that we can all learn how to talk to each other and cooperate, work synergistically. But a Rosetta Stone is not going to cut it, because the Rosetta Stone has only 3 languages inscribed. So we need the Single-Cell ProtEomics meeting. That has more languages. That has math, biology, medicine, mass spectrometry, chemistry, theory, everything I would like it to be. So to make this meeting useful to everybody and to begin to shape this community, I have allocated quite a bit of time to discuss questions that we think are important, as we did last year. So tomorrow, on Wednesday afternoon, we are going to have a discussion in breakout session of questions that you find interesting and important. Here I have listed a few suggested questions, but these are by no means all the topics that we can discuss. So if you have a question that you would like to discuss, I would love to hear about it. Please let me know, and then we can have a breakout session tomorrow where we'll discuss it in a group of people who are interested in that question, and then we'll come together again to hear the summaries from these discussions. Yesterday was a great start of highly interactive discussion. We had so many questions, which I thought was wonderful, and I want to keep this going. I've made sure that breaks are long enough and there is enough time after all the talks for questions, and I would love to hear your questions. Please keep this meeting interactive. The talks are going to be recorded for all talks for which the speakers agree, their talks will be recorded. Of course, if you prefer your talk not to be recorded, we're not going to record it, no problem. But if you agree to have your talk recorded and posted on YouTube, I'll ask you to sign a consent form for that and to bring your presentation slides to the video recording studio at the back of the room so that we can create higher quality video recordings. Last year, some of the videos that we recorded had been seen by many thousands of people, and I've received emails of people thanking us for recording the talks. I think it's a wonderful thing to keep doing it. Of course, I understand if speakers have reasons not to want to record their presentations. All right. So having arranged the logistics, let's go back to the science. And I'll give a very partial response to one of the questions from yesterday. Why measure proteins rather than messenger RNAs in single cells? I'll deal with this question in more details towards the end of my talk, but let me give you one reason. the fate of single cells is regulated by p53 protein. And the messenger RNA for p53 tells us very little about the protein. Why is that? Because we know that p53 is regulated primarily by protein degradation. Is p53 the exception? How about mTOR, one of the key regulators of all cell physiology? Again, you measure the messenger RNA, it's constant across all conditions, all cell types. It's the protein localization, the complex formation, localization, phosphorylation that actually regulates biology. And these examples, prominent examples that we have studied in great detail, are actually not the exception. When we systematically analyzed protein and messenger RNA levels measured across a wide panel of human tissues, we found that in general, messenger RNA levels don't predict particularly well the changes of protein levels across different tissues. They predict very well the overall abundance of proteins, because messenger RNAs for ribosomal proteins are abundant across all tissues, and ribosomal proteins are abundant across all tissues. So if you were to do the naive correlation, you find high correlation, but again, that correlation is not particularly meaningful for the underlying biology, for how lung cells are different from stomach cells. Tells you nothing, this high correlation. So we wanted to do single-cell protein measurements, but most of the available technologies are rather limited. They tend to rely on antibodies, and antibodies have low specificity. Anybody who has worked with antibodies has fun stories to tell of how the antibody wasn't binding the right protein. And we were interested in being able to push to the right and upwards so that we can quantify proteins both with higher specificity and with higher throughput, rather than being limited to analyzing a few dozen proteins per cell, per sample. I found this very useful summary statistic from Nature, an estimate that annually we waste about $1 billion on antibodies that don't bind the proteins that they're supposed to bind. And this is just for purchasing the crappy antibodies. This is not for all the follow-up research chasing incorrect hypotheses because of the low antibody specificity. So we wanted to use a different approach. And one thing that motivated us from the very beginning, and I'm going to come back to this point over and over again, was that proteins are so much more abundant in single cells than messenger RNAs. That surely should help with measuring them. They're over 1,000-fold more abundant. So in particular, being a quantitatively minded person, one thing that I aspired from the very beginning is not just to detect proteins, not just to measure a few copies from each protein, but I wanted to sample enough number of protein molecules from each single cell so that we can quantify protein abundance, not merely detect and say, oh, that protein is present in that cell. I wanted to know quantitatively how much there is. And many of you, certainly those coming from single-cell RNA sequencing fields, are well familiar of the severe challenges of sampling enough messenger RNAs. Even the best methods currently can sample only about 20% of the messenger RNAs in a single cell, which for the median abundant messenger RNA translates to only a few counts. And therefore, even a perfect measurement in the absence of any noise is going to give you a 50% error bar. If you sample proteins 100-fold less efficiently, you get smaller error bars. It's very simple Poissonian statistic. There's no magic to that. And I'm going to come back to this slide. It's a very important slide. So we had a number of ideas of how we might go about to measuring proteins using mass spectrometry in single cells. Ideas that felt very inspiring because they seemed feasible. It seemed like we could actually do these things. And they had a big problem. OK. Somehow I have a different slide. That's OK. I had a big problem. And the big problem was that I had no mass spec instrument. I couldn't do any of that. I had no access to mass spectrometry at Northeastern. And to the rescue came Bogdan Budnik, who provided access to mass spec instruments at the mass spec facility at Harvard University. And together with Bogdan and Ezra Levy, a very dedicated and driven undergraduate at Northeastern University, we started on our first prototype of how we might measure proteins in single cells that we christened SCOPE-MS. We discussed in detail some of the key ideas of SCOPE-MS, so I'm not going to go into that. I'll just mention conceptually two of those. I'll mention the cell lysis. It was very important for us to find a way to lyse the cells cleanly without adding chemicals that are going to interfere with the mass spec quantitation. And the method at the time was focused acoustic sonication by Kovaris. Kovaris got super excited for our preprint because hundreds of people went and asked them to buy instruments from them. But we weren't excited because they knew that this works only because Ezra is incredibly dedicated, holding each tube, one tube at a time, doing the lysis. The volumes were larger than I liked them. So for me, that was a very important stage of the process, but I always knew that we wanted a different method. And of course, our carrier played a very big role. I'll have more to say about setting the optimal level of the carrier. So we had a lot of fun with this. Initially, the question was, can we detect any signal from single cells? That wasn't clear. When we detected signal, we celebrated that. This was such an exciting moment. And then the question was, is this quantitative? Is it just noise? And then we computed some correlations that agreed to expectation, and we celebrated that. It was a really exciting collaboration. And then the Harvard bosses said, no fun without pain. And again, I was in trouble. But I was saved by the NIH Director's Award that allowed me to purchase an instrument for my group here at Northeastern. And finally, I had a mass spec system that I could dedicate to quantifying proteins. And actually, I say I, but it's really these guys here, together with Tony Kohler and David Perlman and others in my group, who made all of this work. It's been a very synergistic, collaborative effort of extracting data from this instrument guided by its developer. So we talked quite a bit yesterday about our current version of SCOPE-MS, SCOPE 2. I'll summarize some of the work. Of course, the first work with Bogdan, the key principles that go into that, and I'll talk a little bit about lysing single cells with our newest method and show you some of the data suggesting that this lysis is extracting proteins efficiently. Very important, very highly technical, but very important thing to show. And of course, you can download all of the data that we have generated and methods. Equally important are the data analytics, because making sense of terabytes of data requires computers and requires doing fairly complex optimization that we couldn't do without the help of statistics and mathematics. And one of the things that has been incredibly helpful for us is that MaxQuant, developed by Jorgen Cox, provides not only quantification of the proteins, but provides a lot of other useful information for the ions that are analyzed by the instrument, information that is incredibly useful for optimizing the instrument parameters. And this is what DoMS, the software package that Gray presented yesterday, is all about. It's using all of the useful quantities in addition to just peptide quantitation provided by MaxQuant to be able to optimize this workflow. And then we have our version of using a very principled and rigorous Bayesian framework to incorporate additional features of the data, such as retention times, drift times, and so on, so that we can increase our confidence in the identification of peptides for which spectra alone do not support confident enough inference. We call this DART-ID, and Albert Chen, who spearheaded its development, is going to tell you about it tomorrow. So, going back to clean cell lysis. We tried a lot of things. We were frustrated with Kovaris. We tried freeze-thaw. Actually, Alex has tried before both Kovaris and freeze-thaw. And freeze-thaw didn't extract proteins well enough. We could detect proteins, no problem, but we could tell that the extraction is much less efficient than extraction with Covaris. We tried a gazillion other things that crashed and failed. I say we, Harrison Speck did that. It's the royal we. And ultimately, we ended up with this very simple procedure of just isolating single cells in a pure droplet of water, a tiny droplet. You can make that as small as you want. We freeze it, we heat it, and then we digest the proteins. Does it work? So the experiment that we use to evaluate this all-important question is a perfect example of the core principle that I aspire to in doing any experiment, controlling for everything that you can. We wanted to have a well-validated method for protein extraction to compare against. And the problem is that all of these methods use chemicals that we cannot put on the mass spec. Now, if we clean up these chemicals, we don't know how much we lost from the cleanup. How do we evaluate extraction independent from losses from the cleanup? Pretty darn tough task, except that you can incur the same losses with a clean sample that doesn't need cleanup. And that's what we did. So we started with light cells that have only light isotopes and cells having heavy isotopes. We mixed the two together. Of course, the light cells were lysed with urea. The heavy cells were lysed with this freeze heat method, which we call mPOP. And then we mixed them together. So we dirtied our clean sample preparation so that we can clean up together the cells lysed with urea and mPOP. And then we looked in the purified peptides, what is the abundance of heavy and light peptides, heavy and light proteins, which is going to tell us specifically what is the efficiency of extracting proteins. And what we found is that across the board, with different durations of digestion, with label swaps, light and heavy, mPOP extracted proteins at least as efficiently or more efficiently than urea. And this, again, held true across different compartments of the cell. Here you can see the results for cytosol, nucleus, mitochondrial membrane, and just about any other compartment that we tried. We extracted proteins more efficiently with mPOP than we did with 8 molar urea. And this result, as I explained, is completely independent from the fact that mPOP is completely clean, doesn't require the cleanup. We did the cleanup as well. So before I jump more into telling you about the biological experiment that we did and some of the things that I'm excited to do with single-cell proteomics, I want to pay attention to one technical aspect of SCOPE MS and SCOPE 2 that has gained more and more interest and discussion around in the community. It is about the carrier and how much carrier should we use. What do you do to determine that? You do a controlled experiment. That's the only thing that's the only kind of experiment worthwhile doing. And that's an experiment that Gray Huffman did to determine, do we need to increase the carrier? Do we need less carrier? What's the optimal amount? So I'm going to show you just a small snippet of data for that. So if you, here the experiments that we did is either using 50 cells as carrier or using 100 cells as carrier. Now, when we use a larger carrier, we can afford to operate the instrument faster because we need to accumulate ions for a shorter period of time. And therefore, we can go fast, analyze more peptides. And that agrees very well with the data. That's exactly what we find. When we increase the carrier by a factor of 2, and therefore decrease accumulation time by a factor of 2, we identify more peptides. That's an obvious way to increase the number of peptides identified. And the signal detected from the carrier doesn't change much because you increase the amount by a factor of 2, you accumulate 2 times less, you accumulate the same number of ions. But what happens to the single-cell quantitation is shown here in this right panel. You can see that the signal drops precipitously, and that's exactly what you would expect. Why? Because you didn't increase the amount of protein from single cells. That's fixed. Now you accumulate for a shorter period of time. What you expect to see is that you have less signal. That makes sense, and that might be exactly what you want to do if you just wanted to increase peptide numbers. But if you wanted to have good quantitation, you can actually play the opposite game. You want to have the longer accumulation times, and therefore much better counting statistics, which I'm going to show later in my presentation. Similarly, you see missing data increasing quite a bit. So here we barely have any peptides that we cannot quantify with high signal-to-noise ratio. When we operate the instrument faster, we begin to see more missing data. Again, I emphasize here that this is exactly what you would expect theoretically by knowing how the instrument operates, and that is really the trade-off. We've never seen any other adverse effects of having the carrier in our system. Nothing that we can measure in a controlled experiment. What we have identified for us is that having 50 to 100 cells as a carrier currently is more than enough to support very confident identification of peptides with the current accumulation times that we use and that give us good quantitation of peptides, and I'm going to show you data to that. So that's an easy way to increase coverage by increasing the carrier size, but you don't necessarily increase quantification of proteins in single cells that way. You increase number of peptides detected and sequenced. So having spent a lot of time developing a technology, we all were eager to use it to answer biological questions. And that's why we're so excited now, because we are getting to that stage where the technology is reliable enough to unleash it to a lot of questions that we have for it. The one that I'm going to tell you today is from the preprint that we just released on looking at factors that drive macrophage heterogeneity. Why is this important? Why does it make any difference? It has been well appreciated for many decades that macrophages in your bodies, an immune cell type, are remarkably diverse. Some of them kill bacteria, as this guy here is doing, and others promote angiogenesis or tumor growth. They have very different, very opposite effects. Some of them are pro-inflammatory, some of them are anti-inflammatory. So we were interested at what drives that heterogeneity. Is it different progenitors? They start from heterogeneous cells to begin with, from different monocytes. Is it different polarizing cytokines that are required for this polarization? Or can you actually get heterogeneity without introducing any of these factors if you start with a homogeneous cell, with a homogeneous population of monocytes? And that is the experiment that we did. We started with again, royal way that Harrison and Ed did. So we started with a population of monocytes, homogeneous. That was a cell line, and therefore fairly homogeneous. One can aspire to have even more homogeneous population, but that's what we did in this case. And then we added PMA, which is just a chemical, not a polarizing cytokine, that induces the polarization of these monocytes into macrophages. And then we selected single cells using FACS sorter, and we analyzed their proteomes as part of the scope 2 design that Harrison talked about yesterday in great detail. I'm just going to underscore here the importance of randomizing the positions of all of the single cells, having the reference, and having the control wells. Without these factors, I think our data would have been barely interpretable, if at all. Again, very important to have controls to know that what you're generating is pattern of signal rather than pattern of noise. Again, yesterday we talked about some of the metrics that we use to determine which single cells would provide us quantitative data. We find that about 80% of the single cells provide highly consistent protein quantitation. Here is determined by low coefficient of variation computed from different peptides, and high enough signal, meaning high enough signal-to-noise ratio from the reporter ions so that we can actually quantify these proteins. 20% is actually 20% lost cells is something that we can easily deal with because this is highly automated. Harrison can produce this in great numbers, these samples, and we can operate the instrument 24/7 for a while. So we are happy with that. Doesn't mean we don't want to improve it. We actually actively are trying to improve it. And Aileen and Barbara sitting over there at the back are collaborators helping us improve it. So we also talked yesterday about the purity. This is my technical slide of metrics that I'm proud, so I cannot not show. We talked about the purity of MS2 spectra as estimated by max quant, the PIF, which is the precursor ion fraction, and whether that measures exactly what is the purity of the spectrum or is correlated to it, it is very clear that it's highly informative about the purity of the spectra. And when we initially started analyzing single-cell samples, the PIF was more like 60% on samples with the same abundance, the same composition. And by MS doing its miracle, we drove this all the way to above 90% consistently across all of the single-cell runs that we did. And here comes the distribution of identifications. How many of the peptide, how many of the ions that are selected for MS2 analysis, MS, result in confident assignment to peptides that can be linked to biology? It's over 40%, and I'm really happy with this number. Again, I'm happy to increase it, and actually Gray Huffman is doing an experiment that I think can bring this to over 90%. But it's a very good place to be. It compares very favorable to most bulk mass spectrometry workflows. Over 40% of our MS2 spectra get very confident, high quality, highly quantitative peptide assignment. And that also goes to the size of the carrier. We don't need to increase the carrier anymore. We obviously have plenty of fragments to be able to assign sequences, thanks to max quant being as good as it is. So what does this translate in terms of peptides identified per single cell? We have on average about over 1,000 peptides that are quantified, not just identified, have high enough signal-to-noise ratio in the reporter ions to be quantified per single cell, which corresponds to about 1,000 proteins. And if we look across a large number of single cells, we see that we have proteins quantified in multiple, meaning many dozens of single cells. We have about 2,000 proteins quantified across many dozens of single cells, and therefore usable for our analysis further on. And at that level, if you do the simple math, we have about 1,000 proteins per single cell, 2,000 in total, so we have only about 50% missing data. The only comes because I'm familiar with single-cell RNA-seq. And this metric, by the way, is the metric that we believe is the metric of number of proteins usable for biological analysis, is the important metric from the standpoint of biological interpretation, because that's what we can analyze. The number of proteins quantified per single cell is a very useful technical benchmark worthwhile keeping in mind. But at the end of the day, if we can increase our throughput so that we analyze tens of thousands of single cells, we might be able to deal with some degree of missing data. All right, so I have been talking about not just detection, but quantitation, delivering. Yeah, Bogdan?
Just go one slide back.
Yeah.
Out of those different graphs, what's kind of your final decision of what you based on your biological differentiation of how much you pick up? For example, for 500 cells, or I don't know, 300 cells, doesn't really matter. Which ones you would work with? Those that have been identified more than 100, more than 50, more than 25, say for 300.
So I'll repeat the question so that everybody can hear it. Bogdan is asking, how many cells do you need to quantify a protein in so that you can use this protein for the biological analysis? It's a very good question, and I think it has specific answers only in the context of biological analysis. What is the problem that you're trying to analyze? And the degree of confidence that you're going to have in the data. You can allow yourself to use proteins quantified in only a few cells to derive conclusions that you should have lower confidence in. Or you may use only those that are quantified across many cells and have higher confidence. So I think the answer depends very much on the question. I can tell you that some of my group members, such as Albert Chen, are extremely rigorous and they don't use any protein unless that's quantified in at least 95% of the cells. That's what he did for our recent paper. I was quite impressed when I saw his degree of rigor. Other people are much less rigorous. They allow a lot more missing data. So I think it's a heuristic. There isn't a simple answer, but ultimately it depends. You have to be mindful of that, I would say. If you use a protein quantified in only a few cells, that's OK, as long as you keep that in mind and as long as you don't make a conclusion based just on imputed data. So going back to this all-important question of delivering enough copies to be able to do the quantification. Because remember, even if we have no co-isolation, even if our instrument measurement is absolutely perfect, our sample preparation is perfect, if we don't sample enough copies, we cannot quantify anything. We are just drowned in counting noise. And thanks to Alexander Makarov's help, actually, providing for us some very useful guidance as to how we can convert measurements in Orbitrap into absolute ion copy numbers, we were able to estimate that for most peptides, we have on the order of between 10 and 100 copies being quantified. And when we group these into proteins, we have about 100 copies of ions being sampled to quantify the proteins. And if one uses a simplistic Poissonian distribution to estimate the counting error, that corresponds to about 10% here based on this curve, and compares very favorably to the counting error incurred in estimating the abundance of messenger RNAs in macrophages, the same type of cells, measured by single-cell RNA sequencing studies. I should also say that these distributions that we show here, unlike the distribution for single-cell RNA sequencing, are not in any way limiting. This is what we have done as part of our attempt to optimize throughput and robustness and decrease cost and so on. If we want it, even with our current LC-MS system, we can easily shift these distributions by at least an order of magnitude further up, increase our sampling, simply by allowing the instrument to accumulate ions longer. That's going to reduce our throughput. That's why we haven't done it. We have to strike a balance. And the 10% counting noise is something that they can live with. I think we can still do science with it. But what I want to emphasize here is that there's tremendous, tremendous opportunity for growth and improvement. That's not a hard wall, sort of the wall that single-cell RNA sequencing has reached. This is just the beginning, our first steps, and we can tremendously increase the efficiency of sampling. Not surprisingly, this sampling affords fairly good quantitation. Yes, question at the back. And actually, I'll ask Sebastian or somebody else to bring the microphone so that everybody can hear the questions. I'm going to bring it for now, but somebody can help me with questions. And feel free to interrupt.
Might be a bit naive, but I was just wondering, in the protein distribution plot, for instance, are you confident that there are no proteins, of functions, say transcription factors that occur between 10 to the 0, 10 to the 1 range, right? Because it sort of flattens out there, or is there a inherent detection limit and so on?
That's right. So what will happen is that we certainly have some ions, some peptides here in this space, and they will have low enough signal-to-noise ratio that we report those as being missing data. Okay. So that's certainly the case.
Well, is it possible, because you show this curve, let's say for mRNA, and there from bulk we sort of know the overall space that we expect, right? So is it possible to extrapolate, what fraction, let's say, of proteins?
No, they're actually intersected. Thank you so much for this question. This is something that they didn't emphasize, but very important. So it is what all of the proteins that we show here are for the same genes for which we show the messenger RNAs. So it is not like we show the distribution for thousands and thousands of messenger RNAs, including lowly abundant ones, and just a few highly abundant proteins. We intersected the two data sets so that we are looking for the same genes. Yes?
Although I have to say that out of those 1,500 proteins, there's still among those the most abundant ones, because that's...
So what Bogdan is saying is that the proteins that we show here are the most abundant ones, and that is correct. In the shotgun mode in which we do the analysis for mass spectrometry, we tell the instrument to go after the more abundant proteins, which is what happens in single-cell RNA sequencing by necessity. What is also important to emphasize is that in the context of scope 2, we can easily change the instructions to the instrument. Instead of telling the instrument go after the most abundant ones, we can tell the instrument go after this set of transcription factors or this set of regulators because that's what they really care about. And this is far more difficult with single-cell RNA sequencing. There you have to design specific primers. You can do it, but you have to physically change your experiment. Here, you just change the instruction on the computer. Much cheaper, much faster. So Harrison already explained yesterday how we compute this correlation here. The important thing that I'll underscore is that bulk measurements cannot be used to validate a measurement in a single cell because they're fundamentally different. If they could be used to validate it, why do single cell measurements? If we are going to take our bulk measurement as being the benchmark, then there's no point in doing the single cell measurement. So what we have done here is we have averaged across hundreds of single cells in silico so that we can get a relative ratio which is representative to the relative ratios that we get from a bulk measurement. We are also interested in being able to do validations on a per single cell basis. But already when we do this validation, actually, it compares very, very favorably to doing the same type of the same type of estimate benchmark with single cell RNA sequencing data. So because the noise in our data is lower than in single cell RNA sequencing, we did not have to filter proteins for our analysis. What is very common these days in the field of single-cell analysis is to limit your analysis only to the most variable genes. Here, we did not do that. Harrison took all of the proteins that we had quantified across large enough number of single cells, and he computed a principal component analysis, decomposed the data into their principal components without filtering anything. And you can see two clear clusters emerging that were then color-coded by knowing whether the cell comes from was a monocyte or a macrophage. And you can see that the labels mostly coincide well with the clusters. You can also see that the macrophage cluster here is more heterogeneous than the monocyte cluster, which suggests that macrophages are becoming more heterogeneous. I'm going to show you more direct evidence for that in a couple of slides. You can also ask what kind of genes are abundant, what kind of proteins are abundant in the cells forming these different clusters. So we color-coded each cluster based on genes that are specific to monocytes or genes specific to macrophages. And again, you can see the clusters being color-coded quite homogeneously based on the corresponding markers. Yeah?
How many out of those 2,321 proteins being detected across how many cells out of those 356?
So the majority of the cell, from these proteins, we are going to have about maybe 2,000 of those proteins are detected across at least 30 or 40 cells. And then about 1,000 are detected across most of the single cells.
And how many differentially expressed proteins you found between the two separate distinct populations?
I'll show you a plot as to that, actually. It's very, so there are two ways, I should say here, to determine the differential expression. One is by statistical significance, and because there are so many single cells, it is very easy to compute ridiculously low p-values. But I prefer to determine differential expression by the magnitude. I not only want to have statistical confidence that the abundances of a protein are different, I also want to know by how much, because you can have a highly statistically significant effect that is utterly scientifically insignificant. Yeah.
Do you worry about the taking into account the absolute value of difference? That, remember, we are working on the field of co-isolation. We know that all our signals are suppressed by default. Just to have too much importance to the value.
I mean, so we can discuss this afterwards in more details. I'm not convinced that we have a lot of co-isolation. We certainly have some, but we have very narrow isolation windows. We have gone to much narrower isolation windows that we used before so that we reduce the possibility of co-isolation. We have worked very hard on the chromatography. Alexander is going to talk more about it. We find that chromatography is all important to reduce co-isolation. So I'm not sure about how much there is, but based on the benchmarks that we have applied and based on what we can tell, it appears that we don't have a lot of co-isolation. Part of that, in this particular case, also has to do with having quantified mostly highly abundant proteins. There are a couple more questions at the back.
Those data actually really highlight why to do single cells, and I was wondering if the macrophage, like each dot is a different macrophage, right? So if you compare the proteins of the one that fall more towards the monocytes, are the protein level kind of similar, or?
That's coming. That's coming.
Okay.
So you're raising a very important point, though. Let me articulate that. There is no expectation in this context that getting perfect separation of the clusters is corresponding to more quantitative data. There are biological reasons why there might be heterogeneity, such as asynchronous differentiation. Some cells are just slower to differentiate. And in general, I think that we should and cannot use cluster separation as a metric for the quality of the data. I made a big point about this yesterday. So you're absolutely correct in your expectation, as the data are actually going to show you in a couple of slides. Very nice, validate your anticipation.
But that being said, the dear monocyte here is like kind of by itself. Would that be an outlier? It seems like they cluster much better than the macrophages. Yes.
We can examine that one and see what's going on in that. You can certainly, one of the cool things here is that we can have reliability estimates on a per single cell basis. And therefore, we can test the hypothesis. Is this monocyte out here because the measurement is poor, or because it's actually somehow more differentiated towards macrophages? We have already removed most of the single cells for which the data appear to be poor, but it could be that this one somehow was left in. We can certainly explore that in more detail later.
Just to go back to the different ways of analyzing the differentiation, the differentially abundant proteins. So for the lower p-values, can you just comment on multiple testing correction? And because in my experience, when doing differential expression, that actually raises the p-values
so what Shiri is saying is that here, if we want to test how many proteins are differentially abundant, we not only have to compute p-values, but we also have to correct them for having tested the hypothesis that very many proteins might be differentially abundant. And of course, that is essential to be done. What I'm saying is that because we have so many data points for each protein across many single cells, we are able to compute very low p-values. And even after multiple hypothesis correction, they remain rather low. So you can have the situation where you have a multiple hypothesis corrected protein with very high statistical significance confidence that it's differentially abundant, but the differential abundance is 5%. Do I want to get excited about this? Maybe if there are a lot of such proteins, but that was my point. So we certainly have to do the multiple hypothesis correction. So the next type of analysis that we did is built upon spectral clustering, which I'm going to attempt to summarize for you very quickly here. It's a very attractive method for analyzing the data because of its strong theoretical underpinning. It's a graph-based method where we can compute a similarity measure between the objects that we want to cluster, in this case, single cells. We can put some weighted edges and have a matrix W in this case that contains all of the weighted edges, connections between the cells. And from that matrix, we can compute the corresponding Laplacian of the graph, which is just a matrix manipulation. What is important for you to know and appreciate, I'm not going to go deep into the math here because we don't have the time for that. What is important for you to appreciate is that the eigenvector of this matrix here with the smallest non-constant eigenvector corresponds to minimizing this objective function here. And if you stare at this objective function, you'll find that it's minimized when cells that are connected by very strong edges, by high weights, also have very similar corresponding entries in the eigenvector. And this objective function can be minimized based on convex optimization, meaning that there are no heuristics. You're certain to get the correct answer very efficiently. And then it's very useful, then once you compute this vector, it's very useful to look into what is the clustering within the cells. And in particular, the thing that we wanted to do here is not assume that we have distinct clusters. We wanted to be able to see distinct clusters if they're distinct, or we wanted to be able to see a continuum of cells if there's a continuum of cells. And when we apply this method to cluster the monocytes and the macrophages to all of the data, what we found is that most of the elements of these vectors were either high and positive, either low and positive, and they actually looked quite bimodal. They resulted in two clusters that are mostly distinct, with a few cells here in between with their protein abundances. And here goes the question for the PCA plot. What about these macrophages that weren't exactly in the macrophage cluster, but were a little bit diffusing towards the monocytes? Well, they're here in the middle, and they actually don't appear to be just noisy. They appear to have a lower level of expression of the proteins that are more abundant in macrophages versus monocytes, and they actually appear to be more consistent with this possibility of asynchronous differentiation. If we use unsupervised agnostic enrichment for biological functions that are overrepresented here in these clusters, we find the suspects. We find that the proteins that are enriched in monocytes, they tend to be involved in cell division cycle proliferation. That makes sense because monocytes are mitotically active. They divide. Macrophages don't. And then if we look at macrophages, we find proteins involved in cell adhesion. That makes sense. Macrophages adhere, monocytes don't. There's suspension. Then we find proteins involved in immune function and so on. So it did make a lot of sense in terms of the underlying biology. And now comes the really cool part. Remember how I told you that this eigenvector doesn't assume what is the clustering, whether you've got two discrete clusters or you have a continuum? So we can just apply the same type of analysis to the macrophage data, and we see a different picture. Instead of having two mostly well-defined clusters, mostly coherent clusters, we see this gradient of heterogeneity. And this is the sort of thing that you absolutely need single measurement for. If we had used markers to sort cells into a number of discrete populations, we would have never known that there is an entire continuous heterogeneity of cell states. If we only had, our data weren't quantitative enough, if we were just doing detection rather than quantitation, we may not be able to see the gradient. The data would be so noisy that we would just lump it into two clusters. So it's a testament to having done single cell measurements that are quantitative enough that we can see this kind of gradient of heterogeneity. Bogdan has a question.
Yeah, two questions. How many proteins on the y-axis here?
So in both cases here, Harrison has chosen to detect the 20% most differentially abundant proteins, which amounts to about 426 proteins or something like that.
And how many of all those 426 was detected across the set? I don't know, how many, 300 cells or 350?
How much missing data there was?
Yes.
Less than 50%.
That's why 50% here is the imputed data.
That's right, about 50. I can give you an exact number, but that would be my estimate.
Are you afraid about the imputation drives these two apart?
No, we are confident that it doesn't, because even if we filter all of the, even if we don't do any imputation, we use only measured values, we still get exactly the same qualitative result, with the difference that you're going to have fewer proteins to display. But we are very confident that this is not caused by imputation. Jorgen.
What is the imputation method that you're using?
Oh, the imputation method. We use k-nearest neighbor imputation, which we know is not the best. We know that needs to be improved, and we are actively working on doing better imputation that is suited for mass spec data. We use k-nearest neighbors because it's the simplest. Barry Carger.
Yeah, so I'm going to ask the question that we discuss. We always worry about in vivo versus in vitro.
Yeah.
And we know well that the cells are affected greatly by their environment, by other cells around and so forth. And so, a fundamental question is, what is the relationship of the results to in vivo?
I'll quickly summarize Barry's point. It's a very important point. When you do single-cell measurements, we isolate single cells, and we can introduce all sorts of artifacts during the isolation. How much of what we see here corresponds to biology versus isolation artifacts? That's a question that exists for all single-cell measurements, and it's much less severe for proteins than for messenger RNAs, because proteins degrade more slowly than messenger RNAs. But nonetheless, we have painstakingly tried to minimize the stress response of the cells. And the way we have done this is by keeping the cells at 4 degrees Celsius. David Perlman, whom I don't know if he is here, when he was in my group, he did a very important experiment to test this effect. And the experiment was to lyse cells on the plate with boiling SDS. So it's physically possible. He took the unsuspecting cells and poured boiling SDS, poor cells. They get lysed on the spot. All the proteins are preserved. There can be no stress response. And then he did the mPOP lysis. He kept it at 4 Celsius for 10, 15 minutes, half an hour. And then we measured the relative protein levels. And we found that, at least in his hands, when he did this control experiment, up to 30 minutes, we actually did not see changes in the protein abundances and phosphoproteome.
Of the environment around the cell, which
Yeah.
when you do your measurements is quite different than in an in vivo situation.
Oh, so you're asking, right. So you can certainly extend the measurements.
Cells are affected by the environment. I mean, that's well known. So.
That's right. That's right. So we've done a biological experiment here in vitro rather than having done that in vivo. Yeah. Yes. Can you?
No, but I mean, just to jump in there, I think it's a very valid point. What I will try to show tomorrow is how to use also imaging data to actually deconvolute this kind of community effect or environmental effect. But at the end of the day, we still need these methods to go there. We need the ability to go in and look at smaller numbers of cells, not necessarily individual cells, from, for instance, a tumor, to deconvolute the heterogeneity, but also the community effects within that system. But that doesn't mean that we don't also need to look at the entire system, because the entire system is often what we want to treat if we're talking cancer treatment.
And that will be a different, very interesting and valid experiment to do. What is important to emphasize here is that having done this experiment in vitro actually has a huge advantage to the in vivo system, because we can answer a question that we could never answer in vivo. What is the macrophage polarization if you start with homogeneous cell population? If we were to do this experiment in vivo, then you're going to start with a heterogeneous population of monocytes. And if we end up with heterogeneity in the macrophages, it is always possible that that just reflects the heterogeneity that we started with. What is really interesting here and got immunologists very excited is that macrophages are able to polarize spontaneously without the presence of polarizing cytokines, without the heterogeneity being there to begin with. But even if you start with fairly homogeneous monocytes, and that is a big virtue of doing the experiment in vitro as compared to having done it in vivo.
Actually, another point is this continuum. We see the same when we deal with cell shape space. So in cell shape space, we also in the past thought this was just down to very discrete, spiky, long cells, small cells. But actually, we also there see continuum. And it's not based on imputation at all, so that support this notion.
All right.
Would it be correct to conclude from the right plot that there are two clusters? I mean, that it's not
Here?
a continuum?
No, I don't think there are two clusters. I wouldn't know where to draw them. Actually, I have a perfect plot for that. Here I'm plotting the distribution of elements of the eigenvectors for monocytes and macrophages. And based on this bimodal distribution, which we obtain in a completely unbiased way, not assuming two clusters, I'm very confident to conclude that here in the entire population there are two clusters. There are the monocytes and the macrophage-like cells. Now, if I look just within the macrophages, we see this continuous distribution. If I were to draw anywhere a threshold, I'll be incurring a big mistake.
But why does it happen then that these profiles once increase and then once decrease in the...
Oh, they're different proteins. So some proteins are more abundant in these cells, and these proteins are more abundant in other cells. So each column here is a cell. So no, I think this is one of the really important conclusions that I draw from this analysis, that sometimes it is incorrect to put the data into clusters. And if you had done t-SNE would have given you clusters, no problem. Doesn't mean that the underlying data falls into two bimodal distributions. Here you can actually see the degree of bimodality, and you can see that the black distribution is nicely bimodal, looks bimodal to me, and the blue distribution is far from being bimodal. Now, what are these proteins that are changing? As Jorgen alluded, some proteins are high in the cells on the left and go down, and other proteins show exactly the opposite trend. So we did gene set enrichment analysis. We overlapped these with genes known to be important in macrophages. And quite gratifyingly, Ed and Harrison discovered that if you look at proteins that are abundant in M1 polarized macrophages, they're high in the cells on the left, and they decrease for the cells on the right. And you see exactly the opposite for genes that are associated with M2 macrophages, meaning that they're upregulated in M2 macrophages, which is suggesting, not proving it, it's not a conclusive proof, but it's suggesting that this gradient here is reflective of heterogeneity that appears to be similar to M1 and M2 polarization, which occurred, remember, without any polarizing cytokines, without any heterogeneity to begin with in the monocytic population. And here is a plot to show you more quantitatively the statement that I made based on the PCA plot, that the monocytes are more homogeneous than the macrophages. We just computed as a measure of similarity between single cells, all pairwise correlations between monocytes and macrophages of the relative protein levels. And we see that indeed the monocytes were much more homogeneous to begin with compared to the macrophages. Yes, Bogdan. How many genes did we use for M1 and M2? We intersected all of the genes that we had quantified in the single cells with the genes that we took from unbiased study, and that was about a dozen genes that intersected. In each case, yes, something like that. You can look at the details in the paper. Question at the back.
This may be a stupid question, but shouldn't you expect for the second eigenvector of a Laplacian to change a sign? So the gradient that you see, wouldn't that be a null model as well?
Would I expect the eigenvector to change sign? This one?
Because the second eigenvector should have some positive entries and some negative entries. That's a guarantee. So
they do. So both the vectors, they do have some positive and negative entries.
What's the null model for the this gradient shape that you have for the eigenvector?
Oh, so here is 0. This one?
Yeah, let's say the blue one.
The blue one, okay.
So if you randomize the protein versus cell association, would you get like a much sharper distribution of eigenvector elements, or?
If I randomize, am I going to get much sharper distribution?
Yeah, maybe. I guess my question is, how do I know that there is signal when you're separating out macrophages by themselves? Because any eigenvector would have a distribution of
That's right.
positive and negative values. So I would like to see if there is a when you sort of.
Oh, so actually, the simplest answer to your question, is this eigenvector significant? Is it just random, or is it telling us something useful for the biological system? This is a question that is well answered with random matrix theory. That's what it's all about, randomizing matrices. And what I can tell you is that when you do this optimization here, the eigenvalue that we get is way, way lower than anything you can get from random matrix theory, which demonstrates that this vector meaningfully captures dependencies in the network. All right. I've had the great pleasure of having a lot of questions, which is wonderful. And that also makes it very challenging for me to finish my talk within the allotted time. But I will go very quickly to tell you about some of the things that I'm very excited that we can do with single-cell proteomics. And I'm going to apologize for rushing through this part because I don't want to take too much time. Now, what we have mostly used single-cell data so far has been to classify cells, to do various cell atlases. And I think that's a worthwhile thing to do, that you can also do with single-cell proteomics, perhaps in some ways better than what you can do with single-cell RNA sequencing. And in fact, we are part with the CZI effort of doing that with these initiatives. But this is not what really gets me excited. I'm not trying to acquire high-dimensional single-cell data only so that I can reduce their dimensionality. I'm trying to acquire highly quantitative single-cell data so that we can do more than dimensionality reduction and cell classification. And some of the obvious things that one can do in the applied realm is, of course, biomarker discovery and precision medicine. But what I'm going to talk more about are two fundamental questions that I'm particularly excited to pursue within my group that go beyond just dimensionality reduction. One of them has to do with post-transcriptional regulation in single cells. We know virtually nothing about it because the post-light has not shown there. We've never had the data to be able to explore what is the post-transcriptional regulation in single cells. I'm going to give you a snippet of very approachable question that is close to my heart that we can tackle there. And the other one is that we might be able to, for the first time ever, to identify direct causal mechanisms in biological data from high-throughput measurements. It's something that we have tried to do for at least 2 decades since the first DNA microarrays. And we've never had, in my opinion, data that could support this kind of inference. We might be getting to the point that we can do that. I will not have enough time now to go into full detail in that. I recently gave an hour-long presentation focused just on that question at the Broad Institute. And if you're interested, you can follow this link here. And you can hear that presentation. I'll give you the executive summary, which I apologize will not be sufficient. Just give you a flavor of it. Wow, that's really the old presentation. All right, so I don't want to take more time. Sorry, I will tell you without slides. Sorry about that. Somehow I got mixed up with my slides, but let me briefly summarize what my slides were going to show. So one of the things that I wanted to show you is that my lab has discovered that there is variability between the protein composition of ribosomes, meaning that cells actually make different ribosomes that could have a role in protein synthesis. And we have done those measurements in bulk samples. So we don't know whether we simply have some single cells making one type of ribosomes, other single cells different, or whether within a single cell we have this variability in ribosomal composition. And that's an experiment, that's a question that is perfectly suitable for the type of measurements that Scope 2 can do. Now, going back to the causal inference question that I think is very exciting, I wanted to underscore that in biology we can very easily infer causality in many cases. If you put glucose on a cell culture, you can measure many messenger RNA and proteins that go up. And if you control for all the other factors, you can conclude that glucose causes these messenger RNAs to go up, these proteins to go up. This kind of causality is easy to infer, but it's also not particularly useful because you don't know what is the mechanism that causes this response. And what has been so hard is to be able to infer direct causality, not just the indirect one that is consistent with a variety of different models, and I go at that with full length in my Broad presentation, but just to infer one or a set of molecules causing another molecular event that is not mediated by something in between. That is one of the big problems of using associations from genome-wide association studies. They give you, even if you strongly believe they're causal, you don't know what is the mechanism mediating that effect. By being able to obtain lots of protein measurements in single cells, we are able to do inference based on minimum assumptions. Basically condition the dependence between two molecules, for example, kinase I and kinase J. We can ask if we choose the subset of cells in which all other confounding factors, all other regulators are the same, except for kinase I and J, you can ask what is that conditional probability distribution between these two regulators. Is their joint distribution simply the product of the marginals, which would imply that they're independent, they don't actually regulate each other? Or is it different from the product of the marginals? And the beauty of being able to do that compared to all of the other approaches that people have tried to do inference is that there are no assumptions of how the confounding variables might do their confounding. You simply control for them experimentally by looking at the subset of cells that are unconfounded. So I think there is a lot of promise to begin to think about this analysis and opportunity to come up with clever algorithms to do it. And I apologize that I couldn't show you the slides. But if you wanna learn more about it, you can follow this link, and then you can hear in more details about my thoughts and ideas about doing this kind of analysis. So I would like to finish by thanking all the people who made the work that I presented possible. It has been a very collaborative group effort, incredibly fun to be part of this journey, to see the excitement at every stage, and to see people synergistically working together towards a common goal, and seeing the progress, seeing how far we have gone starting in 2016 to now in terms of the quality of our data. It feels like a different world. It's been a very exciting journey. Thank you.